Okay, so this is my N209 here at UCI, a behavioral neuroscience course's final presentation. And so we just got to choose a topic from the class that we were interested in and come up with a thought experiment without, too much of a budget or too much constraint to answer our question of interest. And so we studied multiple memory systems and how, like, kind of memory consolidation across different brain regions. And so I'm going to be trying to quantify these remote memories in vivo. And so some background from the class is understanding Tyler and DeSena's hippocampal indexing theory from the 1980s. And so the idea here is that function can follow from form. And so the neocortex, shown here with a bunch of neurons, is a lot bigger, a lot more cortex than the hippocampus. And so during initial memory forming, you will have some amount of perception or features detected about the world by your cortex. And the hippocampus is upstream, listening at a great position at the top of a cortical stack to be able to bind these features detected by the cortex, by a large area of cortex. Okay, and so this will bind kind of in this, it's going up the cortical stack, and the hippocampus will bind all these things into one event. And then initially, right, you need both the neocortex and the hippocampus to retrieve this memory, and the hippocampus will aid in pattern completion, which is essentially like a means of similarity, a measure of similarity. And so we see here that parts of this initial memory are active, and the hippocampus is listening, and in essence can yell down this cortical stack with a different set of connections that go the other way, right? We're going down from high level association cortex down to lower level neocortical areas. And we'll bring back everybody else. So the hippocampus, in essence, go, I'm similar with you guys. You guys usually come with these other guys, I remember, so we'll light everybody up. Okay? And this is going to aid in forming older memories, as we'll see here in a second, because the hippocampus will spontaneously, after you do this initial memory forming, especially like an hour after the learned behavior, if it's relevant learning and not just random stuff necessarily, the hippocampus will spontaneously replay these, even without necessarily a pattern completion component. And this is nice because the cortex, being much bigger, the amount of connectivity in such a big region is not all to all. Every neuron isn't connected to every other region, or every other neuron rather. And this is just a space constraint. I mean, we're wiring things in 3D between billions of neurons if we're talking about us humans. And so by being able to have the hippocampus replay things more than once than the once lived experience, this gives the cortex time to do Donald Hebb's firing together, wiring together. Okay? And so we'll see that here. And so this is kind of what I showed you for new memories. Encoding new memories and retrieving new memories requires this bidirectional interaction between neocortex, hippocampus. But once you've had the hippocampus kind of replay the lived experience multiple times for the neocortex, you no longer need the hippocampus. And there's a lot of synaptic turnover in the hippocampus, many regions that are important for this pattern completion and memory retrieval. So the memory doesn't live here for very long. It can live here silently, but during being an active component, it doesn't seem like it plays a big role for beyond a couple weeks. And what instead happens is what I said here is one of Donald Hebb's cell assemblies. These have fired together enough, thanks to the hippocampus replaying things, that they've wired together, and they support each other. So they do the pattern completion by themselves now. And so this is neuroscience, perhaps the most famous neuroscience patient, HN, Henry Molaison, who had both of his large portions of his medial temporal lobes, including 2/3 of all of his hippocampi, removed. And so he would seem pretty normal at first, having a conversation with him, but he could not form new episodic or semantic memories because of his missing hippocampus. But he had an intact neocortex, so it would take you a while to realize something was wrong with him, because he had a bunch of semantic knowledge. He knew a lot of facts about the world and memories about his early childhood, presumably because they had time to consolidate into his neocortex as independent cell assemblies, not requiring the hippocampus anymore. And so my question and hypothesis is, can we measure these cell assemblies in vivo? If you're familiar with engram work, this seems to complement that very well, because engrams are probably supported by pre-existing cell assemblies in the neocortex, and will ultimately be integrated into a bunch of cell assemblies. But if you don't know what engrams are, that's fine, but look it up. It's fun. So my hypothesis, of course, is, well, we can measure these cell assemblies in vivo. And I think we could use lasers, two different lasers, one to stimulate a small group of cortical neurons, and the other laser of a different light to monitor the resulting nearby activity for pattern completion. And I'll get into it, but this is going to require 2 genetic modifications to be able to have these 2 different lasers cause 2 different proteins to glow in neurons. But the idea would be something like this. We wouldn't know that these connections to cell assembly exist, but we could initially, naively just probe excitatory neurons with our genetic modifications. And in this case, we got lucky, and there is a cell assembly between these 2. So we stimulate them with this laser, and we record all of these neurons with this different colored laser. And actually, there was a cell assembly here. We got lucky, and you see that everybody else got recruited by these 2. And so this isn't, we'll refine this measure. You still might have some uncertainty if this cell assembly exists or not, but like I said, we'll get to that later. We can do better, but just as an initial concept, this is a good way to show cell assemblies in the neocortex pattern completing with our lasers. Okay, and I'm showing my age a little bit here, that I know who Pinky and the Brain are, but my question number 2 is about environmental enrichment. So just having a more interesting environment, more lived experiences, and more semantic memories, presumably. And we think that I think that some forms of environmental enrichment should create more semantic cell assemblies in the neocortex. And this just follows from kind of what I explained, the standard theory system consolidation of memory from the hippocampus initially into the neocortex ultimately. So I think if we can do question 1, if we can measure these cell assemblies in vivo, it follows from standard theory of system consolidation that enrichment should create a bunch of these cell assemblies in the neocortex relative to control mice. So that's Pinky here, who's a little dumb. Okay, and a high-level overview of the experiment. This is our enrichment. We will have a track that the mice will run around to get reward. And the enriched group is running a dynamic track. So an hour a day for 2.5 months. And even within the 1-hour session, the track will be updated 3 times. So like every 20 minutes, some of these will be reversed or turned or swapped places, or completely replaced with new objects. And the control group will be an exercise control. They will just have ramps, like gradual, not too big ramps. That's just more of an exercise control. So that's nice. Not a lot of enrichments necessarily have that type of exercise control. And here's one example of a dual 3D imaging and stimulating laser. So, I mean, there's a lot going on here. I won't go through it all, but, it's just to know, like I said, we've got a stimulating laser of one wavelength, an imaging laser of another wavelength. And here's our mouse with 2 genetic modifications. You have some choices here. GCamp is a very popular imaging genetic modification. So you have CAMK2, which senses calcium internally, and so calcium concentration varies widely when the neuron spikes, the concentration inside the cell. So if you got something like CAMK2, which is hooked up to GFP, and the linker between those 2 is sensitive to actual binding of calcium to CAMK2, that's how you essentially get GCamp. If your laser hits a neuron that's bursting, it'll have a lot more calcium in it. The CAMK2 will be bound, and the GFP will be able to be 40 times brighter because of that binding. So that's how this imaging laser would work. And then Crimson R is a channel rhodopsin. This instead allows you to put light onto a protein that's in there, the far red light, which is nice for deeper penetration into the cortex. And anything that's in the focal plane of this 2-photon laser that has enough of this protein will be excited. Okay. And another thing to note, how we get in this particular, there's several ways to do this, but in this particular 3D imaging setup, there's a piezoelectric objective. So you apply a voltage on here, and it will move the objective up and down, and that will give you multiple Z planes. It'll move it up and down your imaging plane in the brain. Okay, so I introduced you to this earlier. Here is the refinement of how we will tell if we're getting pattern completion or not. Cell assemblies are reciprocally exciting the members in their group, it's thought. When we're randomly stimulating, when we do find something that could be pattern completion like here, if we wanted to further distinguish it from chance excitation, we can change which 2 neurons we're stimulating to another 2 that we saw excite last time, right? And if we get roughly the same pattern with a lot of overlap, then we can feel more comfortable saying that this is true pattern completion. This is we're seeing an actual cell assembly in the neocortex. Where something like this with very low overlap, some different activity than seen in the last pattern, then we probably just say that this is just chance excitation, some off-target activity instead. And since it's known from ensemble papers with similar dual laser setups, that there is different abilities of these neurons to be able to pattern complete, right? Some of them are just able to, like in certain instances, just stimulating 2 neurons. The right 2 neurons can bring back the whole pattern. So there will be variance in this. You will just miss some. They could be part of the cell assembly, but they could be the weaker ones that would require a lot more neurons to be stimulated. But this is just an illustration. A lot of these lasers can actually stimulate up to 80 neurons in a given volume simultaneously. So we can get pretty fancy with this stimulation space. Okay, I got to skip around here. So you can skip we can ignore these first 2 points for now. My second hypothesis about testing if enrichment creates a bunch more neocortical cell assemblies and controls. If we feel good about hypothesis 1, our ability to actually measure these with our lasers, then it's just a simple t-test. You just compare the number of pattern completions, our proxy for cell assemblies, between these 2 groups. So some potential issues is that our measures of pattern completion just might not be accurate enough. So my idea here to give us some ability to refine how we stimulate and detect cell assemblies pattern completing is we could do a novel object recognition paradigm. I didn't find this being used in mice, but I wouldn't be surprised if it was. Mice will, of course, kind of like humans and monkeys, this follows my intuition, is when you show them 2 objects, one familiar and one novel, they will just spend more time around the novel object. And these mice can't move, though. That's the problem. They're head fixed because of these big laser setup. So I just thought, well, if that's their natural response, shouldn't their eye gaze initially do the same thing? And so I found research from humans and macaque monkeys that definitely followed this intuition, that they just stare at these novel objects more than previously encountered ones. Their attention is just grabbed by it, right? And so I think we could do this with the head-fixed mice and show them objects that are new or objects that they saw, if they're the enriched mice, objects they saw during their training on the track. And just maybe measure for a shorter duration than typical object recognition tasks. It could be like 5 minutes for a typical one. So maybe you just do like 30 seconds or a minute of eye tracking on the mice to get a sense of the discrimination ratio. They're probably going to get maybe more bored sooner just because you've restrained them and they can't whisk it or be around it or check the back of it, move around it. And so we can, so we could stimulate any observed cell assemblies this way, but it would only show a subset of all the memories. If we could figure out, if we get enough pattern completion events without going and including this behavior, it'd be nice because you would be able to, presumably, with equal chance, get any of the memories. Maybe that's not true. But as another sanity check, some people have kind of done this similar stuff before. They call them like ensembles, neuron ensembles, instead of cell assemblies. But they seem to be very similar in principle. If you use a different type of opsin for the stimulating laser, so that stimulating them will turn them off instead of excite them, you could actually induce really high error rates in the familiarity scores, just to show you that they're actually supporting the familiarity behavior, if you will. Another potential issue is GCAMP expression, and probably channel rhodopsin too, or crimson R, or the far red variant. They're just variable across time and across cell populations, even when you get them genetically embedded into the DNA as opposed to a viral vector. So even kind of the preferred method has variability issues in expression. And you also have depth working issues. A lot of the ensemble papers with dual laser setups are looking at like layer 2, 3, not necessarily deep into layer 5, which is where you kind of have these even extra huge pyramidal neurons. And the spiking information is limited. It's easier to detect bursts in calcium imaging, but individual spikes are always a lot more difficult, even with all the fancy techniques nowadays. And so you can play along at home. You can try to guess how many actual spikes this neuron has. Give you a second. Time's up. It's around 20. So like I said, these bursts are more easily detectable than individual spikes. So this was an actual probe inside the neuron measuring action potentials. Just happened to have both of these as a sanity check. And then last but not least, this paper and a lot of other papers I've seen with these fancy lasers will stimulate one neuron and then record from others nearby. And I just feel like this is missing precise temporal and spatial relationships that pyramidal neurons depend on in their inputs. So, right, pyramidal neurons are the principal main cell type in the neocortex. So they're very important, and they're very big, right? Sometimes, in the human, they can be like over 1 millimeter long. They can just be ginormous. And so you see a simulation of a CA1 pyramidal neuron here, and this even though it's an old simulation, this very much follows a bunch of electrophysiology, a bunch of electrode recordings, multiple electrode recordings, sometimes up to 3 or 4 in one cell. This follows that real world data very well. So don't worry that this is a simulation. But what this is showing is anything in dark blue, right, which is, almost half of your dendrites. So for a CA1 neuron, I mean, that could be 5,000 synapses, right? So, and, I'm just focusing on the dark blue. This problem, I'm going to say, it actually extends even into the green, right? But the dark blue, any single synapse that comes here by itself, by the time it travels all the way to the cell body, right, it's like a wave crashing on the ocean. It's losing signal, and it loses 900-fold of its signal. It diminishes 900-fold, anything in dark blue, by the time it reaches the soma, if it's by itself, if it doesn't have buddies nearby coming at roughly the same time, sometimes in a specific temporal order. right? And when it does happen, when it's got support from nearby synapses, right? Coincidence support, you can have things like NMDA spikes all over these thin, small, tough dendrites, right? Either the basal dendrites or the far away apical tough dendrites. And these, like I said, are thought to be doing coincidence detection. So these will allow your inputs to actually propagate reliably down to the soma and drive not even individual spikes, but often bursting. These spikes can last for hundreds of milliseconds sometimes. So they are very interesting active conductance properties of neurons that we're missing if we're just recording one neuron and then recording others. You might get lucky you induce an NMDA spike, but I mean the N required for that is probably very large to get significant statistical power. And you also have calcium spikes here at this bifurcation junctions of the big apical dendrites, and even your classical sodium spike here at the axon hillock, right? You have sodium channels all throughout the neuron. And so when you get the spike initiated down here, it doesn't just go down here. It also goes back up into the dendrites, sometimes very far distances. And this, right, you don't even need, we focus on the CA3 pyramidal neurons of the hippocampus a lot for their recurrence. This is recurrence without even axon collaterals, right? The signal you can get feedback just from those sodium channels propagating backwards, and those signals can combine with other stuff coming from other neurons. It's a type of recurrence, a type of feedback inherently most people don't think about. So what does all this mean? It just means that we're already talking about stimulating a bunch of neurons in a small space randomly, looking for pattern completion. That's going to be hard enough, but if we have to get the temporal ordering right, and if we have to get the number of inputs to a single neuron in a small region of dendrites correct, that's going to make doing this even harder and diminish our chances of seeing cell assemblies. And that's it. Thanks for sticking around.